Usually everybody knows that I'm always talking really too much. This time I think I'm going to cut it as short as I possibly can. First of all, I want to thank everyone here. For most part, I think everyone in this room, with maybe an exception of a few newcomers, are all friends of the family of the GIS community, and also all friends of GCRF. So for those who have been around for the 10 years that we actually have fundraised, they basically know that we are very small, simple organization, and basically the only thing that's in our agenda is to find a cure. We don't have any politics in our agenda, and the only thing that we have is just the vision of helping those who so effortlessly really work towards finding a cure. The only way that we are going to be kept alive is by supporting those who are working day and night and so effortless and finding the cure. And those are the researchers themselves that work directly in the labs. And as you, of course, all know, since we are all friends here, you know that GCRF do not get compensated for any of the work that we do. There's no overhead administrative cost, and of course, there's no also overhead administrative cost taken out from the researchers that are working on the research for GIST from the facilities that we donate to. So we want to thank you, first of all. We've been wonderful. You really have been absolutely great. All of you have been part of it, supporting us and making the difference, not only in our lives, but I think making the difference in everyone that is the GIST patients that have been now, but also on the newcomers that are coming behind us. And we are working very closely with the researchers. And for your information also, we have to make another really statement to say that all the researchers that you know of, not only Memorial Sloan Kettering, but all the facilities that we do give the donations, like Pittsburgh, and we give to Fox Chase and OHSU and Brigham and all that. All the researchers really work very collaboratively together towards the same thing, finding a cure. Now you have to remember one thing. All these researchers do not work on repetitious projects. Each one of them has a different project that it makes sense for you to understand that it's not being repeated. And so they really work very well together. They meet together, they talk about GIST, they talk about their work and things like that. So I think that's very important for you to know. I also want to thank two pharmaceutical companies, which is Novartis and Pfizer. We have a few representatives from Novartis. Leslie Fields, why don't you stand up, Leslie? And then we have, was it Tracy? Tracy is also from, and then we have Manny. Manny is from Pfizer. And we thank you very much for coming and being part and joining us in this presentation, because I think you also know that Novartis and Pfizer have been two of the biggest supporters of GCRF and sponsors for us. With that said and done, I want you to know that the pharmaceutical support and sponsorships do not and is not included in the donations that is being done to the researchers. The sponsorship that they provide us with is basically to help us and to bring awareness to and bring the education part in our effort as well. Through our many walks that we have throughout the country, through our international bike ride that we have every 3 years. As a matter of fact, 2012, international bike ride will take place in South Africa. There will be over 25, I think, people that will be from different countries around the world. They will do the 1,000 mile for a cure. And so basically, I just wanted to thank you all for coming and to taking time out from your very busy schedule, because I know you all do. We have Dr. Mark Landsman, who is with the GCRF. You could take a stand. Mark, yes. We also have Alan Statman, who is Esquire, who is the lawyer for GCRF. Alan, take a bow. And so the thing about it is that we want to thank you. We want to thank you not for this year, but for all the years that you've been supporting us. So thank you so very much. We are very happy that Dr. Cristina Antonescu and Dr. Rhonda Medeo is here, that they will be talking about their project and their work and their labs, which is very important to us. And I appreciate their taking time out from their work, clinic, and all the things that they may put aside just to be here for us. So thanks again for coming. I really appreciate all of you being here, and thank you again for your support. Thanks.
Thank you, Tanya, and thank you, Robert, and the members of GCRF for being here, and also for supporting our lab efforts for all this time. I kind of lost track of how many years. I thought it's 7 years, but Tanya may correct me. Maybe it's even more, that you're coming here every year and provide us with a check that we could then go back in our labs and fund our work. My lab people are here, and I see Ron's researchers are here as well. It's very important for them to be here and meet you and sit through this very important presentation. So thank you for being here, and let me just save Mike's presentation very quickly, and then we can start with our agenda. Okay. So just a small modification of the initial agenda that Tanya has circulated on the website. Dr. Peter Besmer could not be here. He's actually sick at home with a very bad cold. He was supposed to give us an update on his very exciting work on the mouse models that his lab is developing on imatinib resistance. However, I think we have a few other very interesting talks, both from the clinical point of view, as well as to update you with what's going on in the lab. So without further ado, I'm going to start giving you an update with what we have achieved from last year, and I'll briefly talk about 3 different items. The first on the exciting ETV1 story. In GIST, this has been a collaborative effort with Dr. Alice's lab from Rockefeller University and Dr. Charles Sawyers from here, published last year in Nature. And then I'll continue with a very interesting study in pediatric GIST, showing you defects in the succinate dehydrogenase, and just give you an example of how the pediatric NIH clinic has brought so many clinicians and investigators together that were able to put together such a big study in such a rare disease, which is pediatric GIST. And lastly, I will briefly update you with our work in progress Because of your generous gift, we were able to study a number of pediatric GIST with the next generation sequencing. So, first I'm going to start with the ETV1 story and how this all came about. We wanted to look at the gene expression profiling across different cancers, including carcinoma, sarcoma, lymphomas, for transcription factors that were previously shown to be involved in cancer, and such as ETV1. And to our surprise, we found that ETV1 was highly expressed in GIST here with a red dot, with very high levels of expression compared to all other malignancies. So then we took this observation and we looked at the gene expression profiling of GIST tumors, and indeed ETV1 came up as one of the most upregulated gene. Here is the top list of genes that is upregulated in GIST. And looking further in a sarcoma database, this is our Memorial database and this is a Stanford database. What really stands out are the GIST tumors, here illustrated in red, showing consistently high expression of ETV1. So the next step was to look at actual tumor tissues for mRNA expression, and here is a group of GIST tumors showing very high level expression of ETV1 regardless of the mutation type, either was KIT mutated or PDGFR alpha or was wild type for these two. And here you can see parallel the KIT expression levels, and ETV1 was not expressed in other sarcomas of the GI tract and was also very upregulated in two GIST cell lines, either imatinib sensitive or imatinib resistant. And this could also be confirmed at the protein level. You can see here a nice Western blot showing a black band here with ETV1 expression really paralleling the KIT expression seen in both GIST tumor samples as well as cell lines. So the next logical question was to address the issue if ETV1 is important in the GIST cell growth and survival. And one experiment that can be done in the laboratory to show that is to knock down that particular gene. And that's what this image shows. ETV1 was knocked down in two different GIST cell lines. Again, one imatinib sensitive, one imatinib resistant. And showing here that the GIST cells cannot grow anymore. If you kill ETV1 in the cells, the cells don't grow anymore and die. Here are two different cell lines, GIST cell lines. You can show both of them show decrease in proliferation. In contrast to an osteosarcoma cell line where ETV1 doesn't do anything. Then, of course, very important is to address the issue, is ETV1 also expressed in interstitial cell of Cajal? Is it a marker of ICC? Interstitial cell of Cajal, of course, we know now that are the precursor cells of gastrointestinal stromal tumors. So these pictures are taken from the mouse gut. And first of all, this is a nice immunofluorescence picture showing that KIT labels the interstitial cell of Cajal in the myenteric plexus of the mouse gut. And actually it turns out that a few years ago, ETV1 was actually shown to be expressed in the myenteric plexus of mice as well. This was a study looking at Hirschsprung disease. So both ETV1 and KIT seems to be expressed in the myenteric plexus, and this is just a diagrammatic illustration to show the different distribution of interstitial of Cajal population in the small bowel compared to the colon. This is again is in the mouse, showing that in the small bowel the interstitial cell of Cajal really group into the myenteric plexus and the deep muscular